Hello, and welcome to our accessible audio-described science videos. I'm Kat.
And I'm Georgia. A black background with white writing that says Georgia and Kat's accessible audio-described science videos with an exclamation mark at the end of the sentence. The words biology core practical 4, osmosis are under it.
Today we are investigating osmosis, which is the movement of water from a high concentration of water to a low concentration of water through a partially permeable membrane, which is a membrane that only some things can go through.
You can now see all of the equipment that is for this experiment, which we have laid out on a table. There are talking scales, a knife and a spatula and a paper towel that are on the right side of the talking scales. A yellow ruler, which is 30 centimetres, is on the left side of the talking scales. A potato with 2 beakers that are 100 cubic centimetres are behind the talking scales, and there are also 6 boiling tubes that are in a boiling tube holder and a container that has got salt in it.
In this experiment, we are using potato strips because they contain plant cells which have unknown concentrations of salt. And we are going to put each potato strip into a different concentration of salty water to see if the water moves in or out of the potato.
There's a whole potato that is on the screen. We have cut off the end of it, and then we have cut one slice of it, which is about 1 centimetre wide. We are then cutting the edges off of the side of the potato so that it doesn't have the skin on it. We then cut the slice of the potato into rectangle strips. We have got 4 strips, but we are going to have 5 different concentrations that are of salt. So we are cutting another slice that is of the potato so that we will get another potato strip.
It is important that the potato strips are the same size as each other because the surface area will affect the rate of osmosis.
We are then putting all of the potato strips next to each other, and we are cutting across the ends of them on both sides so that they are the same length. Then we are measuring how deep that they are with the ruler to make sure that they are all 1 centimetre deep.
The potato has got water on the outside of it that we need to dry off because it will affect the weight of the potato.
We are drying each potato strip by gently rolling it for the same amount of times on a paper towel.
Now we are going to weigh the potato strips, and we're going to write the weight of each in a table, and then we're going to put the potato strip next to the weight of it.
We are weighing the potato strips on some scales, but we couldn't use the talking scales because they weren't accurate enough.
Even though we tried to make the surface area of the potato strips the same, we have still got to weigh them so that we know how much osmosis has happened.
There is a picture that is on the screen that is of a piece of paper that has got a results table on it. The first column is called mass in grams, and the numbers start from 0 and they end at 4. The second column is called mass at start in grams. The weights of the potato strips are 6.08 grams, 6.30 grams, 6.03 grams, and 5.00 grams, and 6.36 grams.
Now we are going to put 20 cubic centimetres of water into each test tube.
There are 5 test tubes that are on the screen that we are pouring water into.
We are going to make different concentrations of salt solution now. The first test tube is going to have pure water in. The second test tube is going to have 1 gram of salt. The third test tube is going to have 2 grams of salt. The fourth test tube is going to have 3 grams in it. And the fifth test tube is going to have 4 grams of salt in it.
There's a beaker that is on the scales, and the scales are on 0, so that all of the weight that is going to be added to them will be because of the salt. We are pouring the salt into the beaker, and then we are pouring the salt into the second test tube, that is for 1 gram of salt, when we have weighed the right concentration of it. We are doing the same things again for all of the other test tubes, for all of the other different concentrations of salt. Then we are using the stirring rod so that the salt will dissolve in the water. But we are drying the stirring rod after each of the test tubes so that we won't add more salt to the other test tubes.
We are going to put each of the potato strips into a test tube. It is important that we know which potato strips that we have put into each test tube. So we are going to add the potato strips in the order that we had weighed them in.
All of the potato strips are sinking to the bottom of the test tubes when we are putting them in there, apart from the potato strip that is in the 5th test tube because it is floating in it.
Now we are going to wait 45 minutes to make sure that there is lots of time for osmosis to occur.
On the screen there is an animation that is of a clock that says 45 minutes later on it. The 5 test tubes are on the screen again. All of the potato strips are now at the bottom of them, including the potato strip that was floating.
After 45 minutes, we are now going to remove the potato strips from the test tubes and we're going to weigh them again so that we know how much osmosis has happened.
We are now going to roll each of the potato strips on the paper towel again to dry them. The salt that is on them is making them stick to the paper towel. And then we are putting them onto the scales. There is a picture that is on the screen that is of a piece of paper that has got a results table on it. The third column is called mass at end in grams. The new weights were 6.44 grams, 5.44 grams, 4.85 grams, 4.06 grams and 5.10 grams.
Now that we have put all the results into a results table, we have got to calculate the change in mass in the potato strips by taking away the mass of the potato strips before we had put them in the water and what the masses of them were after we put them in the water.
The fourth column is called change in mass in grams. The change in mass that is for 0 grams of salt is 0.36. The change in the mass that is for 1 gram of salt is minus 0.89 grams. The change in the mass that is for 2 grams of salt is minus 1.18 grams. The change in mass that is for 3 grams of salt is 0.94 grams. And the change in mass that is for 4 grams of salt is minus 1.26 grams.
We are now going to calculate the percentage change in mass so that we can compare the potatoes even when they started at different masses. To calculate the percentage change in mass, we divide the change in mass by the original mass and then times it by 100.
There is a picture that is on the screen that is of the equation that is to calculate percentage change in mass. The picture is on a white background. The writing that is on it says that percentage change equals change divided by original times 100.
We have calculated the percentage change in mass for all the potato strips.
We are writing the numbers 5.92, that is for 0 grams of salt, minus 14.13, that is for 1 gram of salt, minus 19.57, that is for 2 grams of salt, minus 18.80, that is for 3 grams of salt, and minus 19.81, that is for 4 grams of salt. We have written this in the 5th column, that is for percentage change in mass.
You will notice in the results table that some of the results are positive numbers and some of them are negative numbers. If the numbers are positive, this means the mass has increased and that the mass has decreased if the numbers are negative. The mass of the first potato has increased because there is a higher concentration of water that is outside the potato than there is inside the potato. When the mass of all the other potatoes decrease, it shows there is a higher concentration of water inside of the potato, so the water has moved out of the potato and has gone into the test tube. This is because water will always move from a high concentration of water to a low concentration of water through a partially permeable membrane. Now we are going to put all of the percentage changes on a graph so we can work out the concentration of salt inside the potato.
On the screen, there is paper that has got lots of small squares on it, which is called graph paper. There is a dark line that is after every 5 squares that is across and down on the page. The graph is started by drawing a line that is along the side of the graph paper, and it goes along 30 of the small squares. This line is labelled percentage change in mass, and the sign that is for percent is next to it, in brackets. The next thing that is drawn is a scale of numbers that are for the y-axis, and the numbers start at the bottom with minus 20 and they go up in 5, and the last number that is at the top is 10. A horizontal line is drawn from the 0 that is on the y-axis, and this is to create the x-axis. The x-axis is labelled mass of salt, and there is a letter G that is in brackets that is for grams. At every dark line there are the numbers 1, 2, 3 and 4 that represent how many grams of salt were in the test tube.
Now that we have drawn the axes onto the graph, we are going to plot the percentage change in mass against the mass of salt that was in the solution. The first of the points is at 0 mass of salt and 5.92 grams. I've marked this point with a big cross. The next cross is at 1 gram of salt and minus 14.13 percentage change in mass. The next point is at 2 grams of salt and minus 19.57 percentage change in mass. The next cross goes at 3 grams of salt and minus 18.8 percentage change in mass. The last cross is at 4 grams and minus 19.81 percentage change in mass. We then draw a line of best fit.
The graph points are completed on the screen and the graph is turned 90 degrees. A curved line is drawn through most of the points.
When the concentration of water that is inside the potato and outside is equal, osmosis doesn't happen overall because the water is moving in and out at the same rate, so the mass of the potato will always be the same. This is shown on the graph where the line of best fit crosses the x-axis.
This is being shown on the screen with a cross in a circle, which is labelled no change in mass because the mass is the same. We hope that you have found this video accessible and interesting.
Please feel free to leave us a comment. And don't forget to subscribe for more accessible content.